Welcome to this lecture on the applications of transgenic animals. Imagine trying to build a highly complex, life-saving medicine, but the factory required costs billions of dollars and takes years to build. Now, imagine an alternative where you simply feed and care for a friendly herd of goats, and when you milk them, that exact medicine is naturally present. This might sound like science fiction, but it is a very real and highly successful branch of science today. Let us dive in. To truly understand these applications, we must first formally define what we are working with. A transgenic animal is an animal whose genetic material has been deliberately altered by inserting a foreign gene, known as a transgene, into its genome. Why does this matter? Because DNA is a universal language. This inserted gene becomes a permanent part of the animal's DNA, meaning the animal's cells can read human genes and produce human proteins perfectly. Furthermore, because it is in their genome, they can even pass this new life-saving trait down to their offspring, creating a sustainable source of medicine. Now that we know what a transgenic animal is, you might be wondering why we need to use animals for these purposes when we already have modern medicine and technology. The reality is that human diseases are incredibly complex, and our current medical supply chains simply cannot keep up with the global demand. For example, thousands of people die every year waiting for organ transplants because there are simply not enough human donors available. Furthermore, many human proteins used as drugs are too complex to be made in simple bacteria or yeast. They require mammalian cells to fold them correctly into their functional shapes. Understanding these bottlenecks leads us directly to how we use this technology. The applications of these transgenic animals fall into three major categories. First, producing pharmaceuticals, which turns animals into living bioreactors. Second, generating donor organs for humans, which could completely eliminate the transplant waiting list. And third, creating disease models, like knockout mice, to study incurable diseases. Transgenic animals solve massive medical bottlenecks by providing an abundant, scalable, and biologically accurate way to produce medicines, organs, and test subjects. By tweaking their genetic instructions, we turn them into living, breathing factories. Before we move on to the mathematics of this process, let us see this with a concrete visual example. Here we see the complete schematic of transgenic farming. On the far left, we start with the human gene responsible for a specific medicine. This DNA is carefully microinjected into a fertilized goat egg. The egg develops into a transgenic goat. When this goat matures and produces milk, the milk contains the raw human protein. We then collect this milk and pass it through a laboratory purification column, separating the valuable medicine from the rest of the milk components. The final result is a vial of purified human drug, ready for patients. Now that we understand the physical process of farming, let us look at how we can mathematically quantify its success. When a transgenic animal produces a human drug in its milk, biotechnologists need to calculate the total usable yield to determine if the herd is economically viable. We use a simple but highly practical formula. Y equals V times C times E. Here, Y represents the total usable yield of the pharmaceutical protein in grams per year. V stands for the total volume of milk the animal produces annually in liters. C represents the concentration of the recombinant protein in the milk in grams per liter. Finally, E represents the purification efficiency as a decimal, showing how much protein survives extraction. If the animal produces more milk, V increases, or if gene expression is stronger, C increases, causing the total yield Y to proportionally increase. Let us apply this formula to a real-world scenario. A biotechnology company has developed a transgenic goat that produces human antithrombin, a vital blood-thinning protein. We are given that the goat produces 800 liters of milk per year, so V is 800. The concentration of the human protein is 2.5 grams per liter, so C is 2.5. During extraction, 80% of the protein is successfully purified. We must convert this percentage to a decimal, making E equal to 0.80. Why do we do this? Because it is completely normal to lose some protein during refining, and we need an accurate final number. Multiplying 800 by 2.5 gives us 2,000 grams of raw protein. Multiplying that by 0.80 accounts for the laboratory losses. The final answer is 1,600 grams. This means a single goat produces over 1.5 kilograms of a highly complex, life-saving drug annually. We have just seen how animals can produce drugs, but that brings us to another massive application: creating disease models. Specifically, we create what are called knockout mice. These are mice that have had a specific gene deliberately deleted or inactivated. By removing a gene, scientists can observe what goes wrong, allowing them to study complex human diseases like cancer or cystic fibrosis in a living system. To make these mice, researchers inject modified DNA into mouse embryos. However, this biological process is not perfect. Not every injected embryo will successfully incorporate the new DNA. Because of this, we must carefully track and calculate the efficiency of this transgenesis process to plan our laboratory experiments effectively. To track our success rate in the laboratory, we use a specific formula for transgenesis efficiency. This helps researchers know exactly how many embryos they need to start with. The formula is E sub t equals N sub t divided by N sub I, all multiplied by 100. Let us break down these symbols. E sub t represents the transgenesis efficiency, expressed as a percentage. N sub t represents the number of successfully born transgenic offspring that actually carry the modified gene. N sub I represents the total number of fertilized embryos that were initially injected with the modified DNA and implanted into the surrogate mother. If the laboratory injection technique improves, the number of successful offspring, N sub t, increases. This directly causes the overall efficiency, E sub t, to increase as well. Let us see how this formula works in a real experiment. A researcher is trying to create a knockout mouse model for cystic fibrosis. They microinject the modified DNA into 250 mouse embryos, so our denominator, N sub I, is 250. After gestation, 50 pups are born. However, genetic testing reveals that only 15 of these pups actually have the knockout gene. We must be careful here. We use 15 as our numerator N sub t, not the 50 total pups born, because only the 15 represent a successful genetic modification. We divide the 15 successful offspring by the 250 total injected embryos. This division gives us a decimal value of 0.06, which is the raw ratio of success. We then multiply this decimal by 100 to convert it into a readable percentage. The final answer is a transgenesis efficiency of 6%. Now that we have calculated this 6% efficiency, let us understand what it physically means for the laboratory. This result means that for every 100 embryos the researcher injects, they can expect to get exactly 6 usable knockout mice for their disease research. This relatively low percentage highlights a major reality of animal biotechnology. Scientists must inject hundreds of embryos just to guarantee they get enough transgenic animals to establish a stable breeding colony. It is a highly labor-intensive process that requires immense precision and patience. However, despite the low efficiency, the resulting disease models are absolutely invaluable. Without these specific knockout mice, finding cures for devastating genetic diseases would be nearly impossible. We have covered a lot of ground today, so let us review the key takeaways. First, we learned that transgenic animals have their genetic code carefully modified with foreign DNA, allowing them to produce complex human proteins. Second, we saw how this transforms them into living bioreactors, providing a scalable way to produce life-saving pharmaceuticals and even donor organs. Third, we used the yield formula to mathematically prove how efficient and valuable this farming process is. Finally, we explored how knockout mice act as precise models for incurable diseases, and we calculated the transgenesis efficiency required to create them. You now have a solid mathematical and conceptual understanding of exactly how and why we use transgenic animals in the real world. Great job!